Good afternoon, everyone. My group will be presenting on a topic titled Phylogenetic Relationships and Bioinformatics. My group consists of 7 members, namely Yugutipuram, Kuksam Bakas, Jamie Singh, Louisiana Dirup, Fondel Austin, Trevor Tobin, and myself, Ravindra Achodio. Now, here we have the outline of this presentation. Beginning this presentation, I'll be answering the question, what are phylogenetic relationships? Now, a phylogenetic relationship basically refers to the similar characteristics that are shared between uh, a group of species or species and its common ancestors. Now, those characteristics can be genetical as well as physical characteristics. Now, here we have an example of a phylogenetic tree. There's a very simple tree, and if you were to look at this tree here, based on time 1, time 2, species A, B, and C, we can clearly see that species B and C are very closely related due to the species B and C having a more recent common ancestor. However, species A, though it may not be as closely related as species B and C, species A is still related to those two other species. Now here we have another example of um, three phylogenetic trees. Now in this example, it's basically showing you that um, there can be a difference in the different phylogenetic trees. Now looking at the first phylogenetic tree on the left, and looking at the phylogenetic tree in the middle, you can clearly see that the 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 tree on the left and the tree in the middle, they look identical and they look almost as if they were mirror images. However, if you were to compare the phylogenetic tree one and the second one to the third one, you can clearly see that the third one has uh, it looks different basically. So what this basically tells you that um the phylogenetic tree one and phylogenetic tree two are basically equal, but the third tree indicates that there is a different relationship. I will now hand you over to the next presenter that will take you through the other aspects of this presentation. Thank you.
Good afternoon, everyone. My name is Yugita Putram, and I'll take you through how phylogenetic relationship can be established in bioinformatics. First of all, in bioinformatics, we need the DNA or the protein sequences of the particular organism, group of organism, or species of which we want to find out the evolutionary information of. And so the DNA are the protein sequences. These are molecular data that are useful in providing the evolutionary perspective of the existing organism. And so the genetic materials, over time, they accumulate mutations. And so mutation are responsible for the phenotypic changes over time within a group of organism, a family, or a species. Now, the genes in this case, the genes serve as a molecular fossil, and so they are the medium for recording the accumulated mutations. And so, in bioinformatics, in order to find out how or where or what are the evolutionary changes, comparative analysis is used of the molecular fossils. And so, and this is done for several related organisms. And so the evolutionary history of the genes and even the organism, it can be revealed through the comparative analysis process. And so this is done with some development of the bioinformatic tools. Now, bioinformatic tools, we'll get down to that later in the presentation. These bioinformatic tools, they help to analyze large data sets in practical computing, and so they yield an optimal solution with high probability. So these bioinformatic tools, they're very helpful in analyzing large sets of data, and so we can get information on the different evolutionary changes that occur in a number of species or a group of organisms. Now, as I mentioned, the comparative analysis that takes place in bioinformatics to find out the evolutionary changes of these organisms or the phenotypic changes. So the steps in the phylogenetic analysis, first of all, you select an organism or a gene family. So organism or the family with desirable traits and characteristics, we select uh, the gene from that family, and then next step is you choose an appropriate molecular marker, and so that the then we amplify, sequence, and assemble the gene, and so this is done by identifying a protein or a DNA as mentioned before. And so the DNA sequence of interest, it can be retrieved using the NCBI BLAST or other search tool. In bioinformatics, you have a number like the cluster of W and so forth. And then once the sequence are selected and retrieved, we create multiple sequence alignment. And so this involves arranging a set of sequences in a matrix to identify the region of homology. And so there are many websites in bioinformatics that can be used to retrieve these information, or it has multiple sequence on given set of molecular data. And some are like the MSA and so forth. Then we we have the evolutionary model. And so the evolutionary model is where we build an or estimate the phylogenetic trees. And so from this is built from the sequences using computational methods or stochastic models. Then we have phylogenetic analysis. Now phylogenetic analysis is done by using one of the bioinformatics tool. And so the tool helps to gather information on the different phylogenetic changes. And then we have the tree construction. And so to build a phylogenetic tree, statistical methods are applied to determine the tree topology or calculate the branch lengths and so forth to describe the phylogenetic relationships. And so the most computation method applied to the tree construction, we have distance matrix method or the discrete data method. And so there are several softwares that can also be used in this method. Then we have the evaluation of the phylogenetic tree. Now, the tree statistical tests and assesses to the estimated trees are done. And so tree estimating algorithm generate one or more optimal trees. And so the set of possible trees is subjected to a series of statistical tests to evaluate whether one tree is better than another or and if it the proposed phylogeny is reasonable. Now I'll hand you over to the next presenter.
Good afternoon, everyone. My name is Louisiana, and I'll be taking you through bioinformatics tools that are used in phylogenetic analysis. So firstly, we have PANTER, and that is an abbreviation for Protein Analysis Through Evolutionary Relationships. And what this is that it is basically a comprehensive database of protein families, trees, subfamilies, and functions. Also, one of the main applications of PANTER is an accurate prediction of the functions of uncharacterized genes that are based on their evolutionary relationship to genes with function that are known from experiments. Also, the PANTER database tend to provide phylogenetic trees of protein coding genes. Each gene family tree is annotated with the evolutionary events that explain the tree topology, such as speciation and gene duplication. Secondly, we have the PPOD tool, and that is abbreviation for Princeton Protein Ontology Database. And this is basically a database system that allows users to find and visualize the phylogenetic relationships among predictive orthologs. And orthologs are basically a gene in two or more species that have evolved from a common ancestor. So this tool visualizes the phylogenetic relationship among predicted orthologs to a query gene from any of the 8 eukaryotic organisms, and to see the orthologs in a wider evolutionary context. Also, the PPOD, it offers a simple way to visualize and analyze the relationships between homologous proteins in order to infer function. Thirdly, we have PFAM, and this is basically a database of protein families that includes their annotations and multiple sequence alignments. The general purpose of this tool is to provide a complete and accurate classification of protein families and domains. And now for each family in the PFAM, one can basically view a description of the family, look at multiple alignments, they can view protein domain architectures, they can examine species distribution, follow links to other databases, and also view known protein structures. Fourthly, we have TreeFam. And TreeFam basically aims to be a resource for identifying orthologs between animal species and for studying the evolution of animal gene families. In TreeFam, orthologs, which are a gene in two or more species that has evolved from a common ancestors, and paralogs, which is one of a set of homologous genes that have diverged from each other as a consequence of genetic duplication. So basically, in tree-fam, orthologs and paralogs are inferred from the phylogenetic tree of a gene family. And lastly, we have phylofacts, which is a structural phylogenomic encyclopedia. And this encyclopedia contains almost 10,000 books for protein families and domains. They consist of pre-calculated structural, functional, and evolutionary analysis. Other functionality of this that are provided by this tool includes prediction of protein structures, active site residues, and cellular localization.
Good afternoon, everyone, and I'll be taking you to the applications of phylogenetic analysis. Firstly, it can be used in the medical and biological fields. As it relates to the medical field, one example is that it could be used to determine the evolution and the diversification of tumors. And as it relates to the biological field, it can be used in forensic sciences, conservation biology, epidemiology, drug discovery and drug design, etc., etc. I'll now hand you over to our other presenter. Thank you.
For conclusions, firstly, we know that phylogenetic relationship, it tends to refer to the relative times in the past that species tend to share common ancestors. Also, phylogenetic analysis is known as a study of the evolutionary development of either a species or of a group of organisms or even a particular characteristic of an organism. And this is usually done in a series of steps. There are various bioinformatic tools that can be used in phylogenetic analysis in order to provide efficient information on an organism or a species. And lastly, phylogenetic analysis can be applied in a number of different fields in bioinformatics. These are our references, and thank you very much for listening.